All right. Hello, everyone, and thank you for joining us today. My name is Megan Weber. I'm an extension educator with the University of Minnesota's AIS Detectors program, and we're excited to welcome Carly Wagner here for today's webinar about starry stonewort impacts and spread in Minnesota. I'm going to give a couple of quick housekeeping items, and then I'll allow Carly to get started and introduce herself. So everyone is going to be muted for the duration of the webinar. If you have questions, you can see the image of what you should see at the bottom of your screen with the chat window. Go ahead and type your questions in there. We'll be monitoring that and pulling them. So go ahead and enter them at any point during the webinar, and we'll pull them and keep tracks that we can start answering questions at the end. If you have any technical issues, go ahead and email Pat Mulcahy. You can see his email address on the screen now. And with that, I think we're ready to turn it over for Carly to introduce herself.
All right, hi everyone. Thanks for joining me. Thanks for the introduction, Megan. My name is Carly Wagner. I'm a graduate student here at the University of Minnesota Twin Cities, and I'm also affiliated with the Aquatic Invasive Species Research Center here on campus. And I'm a master's student, and today I'm going to be talking about my thesis research that I did on starry stonewort. Throughout my higher education, I've got to work on various projects on starry stonewort, from phenology to the drying tolerance of starry stonewort and some in situ management. So I've been involved with a lot of projects on starry stonewort, and I'm really excited to be sharing the results from my thesis research on the impacts of this species in Minnesota lakes and how it spreads in our lakes as well. And I'm shortly finishing up my thesis here, so I'm looking forward to sharing these results. I've only presented them once, so thank you for joining me, and you'll definitely learn something new about this invasive species. Okay, to make sure everyone's on the same page here, let's introduce the star of the talk, why we're all here today, starry stonewort. There's some few things that I think are the main points to know about this species. It's a green macroalga. It's not an aquatic invasive plant like curly-leaf pondweed or Eurasian water milfoil you might be familiar with. So it has a little bit of a unique biology and ecology, even though it kind of looks and behaves and grows like a traditional aquatic plant. The second important thing to know is that starry stonewort has these star-shaped reproductive structures. These are for vegetative reproduction. So one of these star-shaped bulbils can grow into a new individual of starry stonewort. These are really important for its growth and spread in its invaded range of North America. And another reason to know about these is that it's the most identifying characteristic of starry stonewort. So if you see these star-shaped bulbils, you can be confident this is the non-native macroalga. And it's important because we do actually have a lot of native macroalga in Minnesota and also in North America. We call these charophytes, chara or muskgrass. So they can be difficult to distinguish between the invasive starry stonewort and our natives. But if you know about these bulbils, then it's a good trick to determine. One of the reasons we're so concerned about starry stonewort, why it's so invasive, is that it can form really dense mats. You can see in the right photo there, basically just a full blanket of starry stonewort. And it's really easy to see when you're out on a lake. It's just a carpet along the lake bottom. So that's why one reason we're concerned about this invader. So starry stonewort in Minnesota, we've only known of it in this state since 2015. It was first identified in Coronis, a central Minnesotan lake. And since 2015, it's been identified in new lakes each year. And in 2019, currently, it's in 14 lakes across 6 counties in the state. So since we've learned that it's in the state, it's had a growing presence. And just last year, the first metro lake was identified with starry stonewort, which kind of elevated the threat of starry stonewort spreading in Minnesota because metro lakes are very high use. There's lots of connectivity to other lakes in the state. So for the last few years, it's been a growing problem in Minnesota lakes. But what do we know about this invasive species? What's it doing in our lakes? And how much should we be concerned about it? So for a species that's growing in presence in Minnesota and also in states like Michigan, it's widely established. Indiana has some populations. It's a growing concern in Great Lakes, but what do we actually know about it? To be honest, there's not a lot of research. In the last few years, publishing information has been coming out more and more, so we're learning more as we go. But one thing we do know about starry stonewort is that it has recreational impacts. If you've seen it out on a lake, it can grow in really dense mats that take up a large volume of the water column and in shallow water, even reaching the surface. So if you have that on your lake shore or around a boat axis that you use, it can definitely impede use. And that's one reason to be concerned about it, our recreational impacts. But one of the key things to determine with an invasive species are what are the ecological impacts. You know, we care about invasive species because when they come to native habitats, they can have a big impact altering the species that are there, the processes in those ecosystems. So that's why we're concerned. And for starry stonewort, its ecological impacts are still mostly a big question mark. There's been some work done, but no work on the impacts in Minnesota lakes. So we're not sure if starry stonewort is going to coexist or commingle with native species, like shown in the left photo. There's a carpet of starry stonewort on the bottom, but you're still seeing some nice native milfoils sticking out of that mat. There's still native organisms using that habitat, like fish. So we don't know if it's going to be that type of scenario where there's a lot of commingling. That'd be good, a positive thing. Or if starry stonewort is so competitive and aggressive that where it occurs, it just kind of forms a monoculture where it's just starry stonewort, a blanket across the bottom. And you can see why it'd be important to figure out which of these two states or a mix of these two are going to occur. You know, if it's in your lake, you're going to be concerned about what are the fate of the native plants in that lake and how is my lake going to look in a few years. So to fill this knowledge gap, I did my research on the impacts of starry stonewort, and the objectives of my research were to first assess the impact of starry stonewort on native aquatic plants. We care about our native aquatic plants because they're a good source of diversity in our lakes, and a healthy native aquatic plant community has been shown to have positive effects on other taxa in lakes like fish and invertebrates. And another aspect of my research was to quantify how starry stonewort invasion progresses over time. So once we get an idea of what are the impacts of starry stonewort, we can try and look at over time, what is starry stonewort doing in our lakes. So the first part of my talk is going to be going over looking at starry stonewort's impacts broadly across 3 Minnesota lakes. And then I'm going to move into the second part of my talk, which involves multi-year sampling, focusing in on starry stonewort in 2 Minnesota lakes. The goals of my research were to reduce uncertainty on the threat of starry stonewort. As I said before, we're not quite sure what starry stonewort is doing in our lakes once it invades, and that knowledge gap needs to be filled in order to effectively manage this invasive species. And then if we get a better sense of what are the impacts of starry stonewort, that can inform prioritization of species management. And if we find that it's a very high impact invader, then we're justified in doing the outreach and research and continued management on the species that's currently ongoing. So now getting into my research, where did I do this? Looking at starry stonewort's impacts. I chose 3 Minnesota lakes that have starry stonewort, and these were good candidate lakes because they all have broad enough infestations to sample extensively in those lakes. Like I said, there's about 14 Minnesota lakes that starry stonewort is currently present in. But a good thing is that in a lot of those lakes, it's contained to a very small area. So these 3 lakes that I chose already have a fairly broad infestation of starry stonewort. And another good thing about these study lakes is that they're representative of a diverse types of lakes and also native plant communities present. So we can try and see if the impacts of starry stonewort are similar across lakes or if they're more unique to the community and lake in that system. So Coronis is a central Minnesota lake. It's an agricultural dominated region, semi-eutrophic, fairly low to moderate species diversity. And then Winnipegashish and Moose are more north central Minnesota. These are low nutrient lakes, fairly high diversity. And then Winnipegashish is a flow through for the Mississippi River. So that has a little bit of a unique community there as well. So now I'm going to tell you how I did my research. I'll show you this video first and then describe it. So here's a meter tape along the bottom, and that's all starry stonewort on the bottom. And then we're moving into now native dominated habitat. So just get a sense of what I was kind of looking at while I was doing this research. So the idea for this research, we definitely wanted to use scuba-based sampling. You know, a lot of aquatic plant research is boat and rake toss based, and that's really good for getting broad population assessments of native, aquatic, and invasive plant communities. But since I wanted to look at impacts and how starry stonewort is interacting with native species, using scuba and transect design is the best for that, because we can be sure we're detecting species that might occur at low densities or they're more rare. And then we can also be sure that we're consistently estimating the abundance of species accurately. So these transects were set up to start in one end that had a lot of starry stonewort, as you saw in the beginning of the video, and then the transect would extend about 25 meters into an area that has a lot of native species. And the idea with this sampling design is that we can look at how native communities change as starry stonewort abundance gets higher or lower, and look at those differences to try and make some inferences on how does starry stonewort potentially impact native communities. When there's lots of starry stonewort, like in the first half of the transect, what species are there and how much? And as we move into an area that's uninvaded, what are we seeing there? So here's a photo of what this sampling design looked like. We had the transect set up along the bottom, and then we used a 1 square meter PVC quadrat frame, and then sampled and collected data within that square meter. And we did this all along the transect. So we'd have about 25 meter transects, and we'd just move the quadrat along those transects and record what we saw. What data we were recording was what species were present. So we identified all native and invasive aquatic plant species in the quadrat, and then estimated their cover and how much of those species were present. So the reason I used cover to determine the abundance of aquatic plants in this study is because it allowed me to sample broadly across the lakes. Some research you can use biomass, which is a good, of course, indicator of abundance, but that's really intensive and I wouldn't have been able to do as extensive sampling if I did biomass. So I used cover as an indicator of abundance. And this was a cover class scale, so it goes from 0 to 8. 0 was absent, and then 8 was 100% across the quadrat. So here's another shot of what this looked like. So here, if I was on top of this collecting data, I'd see starry stonewort is covering that entire quadrat. I'd give that an 8, it's 100% cover. But then we also see another species there right to the left of the transect tape. Looks like some type of pondweed covering not much of the transect, so I'd give that a 1. It's a 0 to 1%. So this gives you an idea of kind of the sampling design that I used. And in each of the 3 lakes, I had about 25 transects each. Using this data, I was able to look at how starry stonewort is associated with the cover of native species, with the number of native species or richness, and then I also looked at impacts to individual species and groups of species. So total cover and richness are the most commonly assessed responses in invasion impact studies. One, because they're easy to sample, it's a quick measurement, and they're also very good general descriptors of the overall health of the native aquatic plant community. You know, how much native biomass you have is positively correlated to how many ecosystem services are provided. And then diversity or richness we value for its intrinsic value, but it also can help with resilience and improved habitat for other species. So I was glad I was able to look at total cover and richness, and because I had a fairly large data set, this was quite intensive sampling, I was also able to look at impacts to individual species and groups of species. And this is good because we have fairly good diversity of species in Minnesota lakes, so it's important to figure out which ones are being affected. You know, it's likely that species aren't all going to respond the same to one particular invader. And then looking at groups of species, and what I mean by this is, you know, how do they grow? So are they unrooted? Are they tall? Do they have floating leaves? Looking at that type of morphology group, and this gets at kind of the mechanism of impact. You know, if starry stonewort is affecting plants that are rooted, potentially it's taking up space. So it gives us a little bit better idea of maybe what's the mechanism of impact. And this is also good for generalizing the impacts of starry stonewort to other lakes. So now we're getting into results. From this transect data I just described to you, let's start by looking at the axes. On the x-axis, I have Nidolopsis, which is starry stonewort cover, from 0 to 8. Remember, 0 is absent, and then 8 is 100% cover. And the y-axis, we're looking at native cover. So in that quadrat, what's the total cover of native species? It's grouped by all the species that are present. And that follows the same class. 0 is absent, 8 is 100% abundant. The gray dots you see on this graph are individual quadrats. So you can see we have a lot of quadrats that we sampled, and looking at all this data, we're able to fit a line of what's the relationship between starry stonewort and native cover. So you can see there's a really strong, significant relationship. As starry stonewort increases, you have a steep decline in native cover. And it's important to point out that this cover class system is additive, so it can be more than 100%, because if you have a species growing low on the bottom, that can have a 50% score, and then you can also have species that are taller, taking up space in the canopy. So we're seeing a strong negative relationship despite that. As starry stonewort increases, we're having a lot of decline in native cover. And the 3 different lines are my 3 different study lakes. We're seeing a consistent trend across different lakes. If you're having a hard time picturing what this graph looks like in the real world, on the top left we have what we'd call 100% native cover. It's lots of diverse heterogeneous habitat. There's things at different heights. You can see there's different species. And then on the bottom right photo, you can see that's just 100% starry stonewort. So this is kind of the differences we're seeing from uninvaded habitat to habitat that's really heavily dominated by starry stonewort. Now moving into what is the effect on the number of native species present. So in a quadrat, how many native species are there? That's now what is on the y-axis, it's native richness. And the x-axis remains the same, just starry stonewort cover. So here you can see it's a little bit more slight relationship. This was significant that as starry stonewort increases, native richness declined. And we saw about a loss of 1.5 species was from uninvaded habitat to habitat where starry stonewort was 100% cover. So 1.5 species might not sound like much, but remember this data was collected at only 1 square meter scale. So if you're seeing losses about 1 to 2 species at that scale, think about how that would extrapolate to all the area that starry stonewort might be covering in a lake. So we're seeing a decline in native richness, but there's more to learn. All these dots represent, 6 species. We want to know what species are there, who's maybe dropping out as starry stonewort gets more abundant. So that's why we look at impacts to individual species. And for this, I just fit models to starry stonewort abundance. How does it affect each individual species that I found in this study? And if you can familiarize yourself, maybe you'll see some plants you're familiar with. We found 30 species across this entire research study, and 11 out of 30 species were negatively affected. And I think one of the most important players to be aware of is the native charophytes, because native charophytes are sometimes native dominant in these lakes, especially in Coronis, which I sampled. So if starry stonewort is really displacing or negatively affecting those species, that could be a big change for habitat. And also, native charophytes are a fairly diverse group. There's at least 4 or so common species, and then even more less common, but it's a good diverse group. So if one invader, starry stonewort, is negatively affecting all those species, that could be a big loss of species overall. So we know generally a big group of species is likely affected, but it's kind of mixed. There's some pondweeds that the data showed were affected, pondweeds that weren't affected. So this is why we get into a more lumping analysis, the one that's potentially more generalizable. So I grouped the species that I found into different morphology or growth form groups. For example, the carpet group is groups like caras and naiads that are really low growing, that grow really similar to starry stonewort. There's pondweeds, that's a very large group, and then groups that are unrooted, like macrorrhiza, the bladderwort species, and coontail. Potentially those could do better with starry stonewort because they don't have to be rooted. So I wanted to look at this growth form analysis to try and get an idea of maybe why is starry stonewort impacting native plants. And what I found is that the groups that I tested, most were negatively affected. The only group that wasn't affected was floating leaf species, which makes sense because they're kind of single-stalked plants that shoot up to the surface really quickly and then put out floating leaves. So potentially those species don't have to deal with this big mats of starry stonewort as much of these other species that are growing on the bottom closer to starry stonewort. So I looked at all these different native responses, the abundance of native species, the number of native species, and then looked at individual species and growth form groups to try and get a sense of how well does starry stonewort play with native plants or not. And our observations point to no. Starry stonewort is associated with negative impacts to native plant communities for all these responses. But the caveat of this data set is that we're not showing direct impacts. We're not showing that starry stonewort displaces native species. This data is looking at differences across space, across the transect, to try and tease out the impacts of starry stonewort. Remember, we're looking at where starry stonewort is abundant and not abundant, and looking at the differences in native communities to try and say something about how starry stonewort is associated with native plants. So to try and get at this idea of determining what are the direct impacts and looking at how starry stonewort is directly affecting native plants with absolute certainty, this is where my second objective comes in, which is quantifying how starry stonewort invasion progresses over time. And instead of sampling broadly across 3 lakes at a single point in time, I had transects set up in 2 lakes that I was able to revisit over multiple years to see how starry stonewort invasion grew over that time period and what happened to the native community. So I called these my resampled transects because we're revisiting them year after year. We're using the same sampling design where transects are set up from invaded to uninvaded habitat, except now these ones are permanently marked with stakes. So we're able to restring the transect tape on the exact same path of vegetation each year. So these resampled transects are set up in Moose Lake and Coronis Lake. They were set up in 2017. Each lake has 15 of these transects. And I'd like to thank Dr. Ranjan Muthakrishnan, who kind of initiated the setup of these transects. So we sampled these transects in 2017. We revisited some of them in Coronis in 2018 and tried to revisit all of them in 2019. So I'm going to show you what happened on these transects over time. I'm going to preface how I'm going to present this data before I get into all the graphs. So I'm going to show you on the x-axis is meter points along the transect. So think about that x-axis being you're progressing along the transect, and each little tick mark, what was the native community and what did starry stonewort look like at that point. And I'm going to show starry stonewort in red, and the axis for that is 0, starry stonewort's absent, all the way up to, again, 8, where starry stonewort is 100%. And then I'm going to show you native richness right next to that. So we're reading that as a simple count. The bar graph's at 4, there's 4 native species at that transect. And I'm going to show you the sampling effort over time. So here's year 1, it moves into year 2, and then year 3. So one graph is one single transect that we were able to track over a 2 or 3 year period and see what changed in that time. I'm going to present Coronis results to you first, and then Moose Lake results. So here's 8 of the Coronis transects. moving from year 1 to year 3. You can see over this 3-year period, Starry stonewort pretty much took over all of these transects in Coronis Lake. And when we're seeing Starry stonewort increase, we're seeing a decrease in native richness. So now we can say Starry stonewort grew very abundant on these transects, and in the same time, native richness declined. So we can be very confident that that's likely a direct impact of Starry stonewort. And now in year 3, when we sampled them this year, basically Starry stonewort is covering the entire transect. It's just a monoculture mostly of Starry stonewort. And if you need a refresher of what this looks like, think a mat of Starry stonewort just extending across the whole transect. So maybe in 2017, we'd see these native species. When we revisited the next year, there was less, and then in 2019, it was all Starry stonewort. So it was a big change over just a 3-year period that we were fairly surprised about. Now we're moving into our different study lake, Moose Lake. And I'll give you a second to watch these. These were only sampled in year 1 and year 3. And what we're seeing in Moose Lake, it's not as abrupt of a transition as we saw in Coronis. You know, Coronis across all transects, Starry stonewort basically took over and we lost a lot of native species. In Moose Lake, it's more mixed. Starry stonewort declined in some areas, got a little more abundant in some, and we saw fairly good native presence between year 1 and year 3. So this was a little bit different pattern than we saw in Coronis. Potentially Starry stonewort not being as aggressive in moose, maybe because there's higher diversity in that lake, we're not sure. But one of the caveats is that we saw a good mixed native community on most transects, kind of like in this photo I show you here. Starry stonewort was present, but there was still native species. However, there were 2 sites in Moose Lake that we were completely unable to resample because the Starry stonewort was so abundant. It was probably 5 feet or more thick mats, and we just weren't able to relocate our stakes to resample these transects. So there was 2 sites or 6 transects where we sampled them in year 1, and the Starry stonewort just became too abundant in the 3rd year, where now we can't see what happened with those transects. So while we saw mostly native communities holding on fairly well in moose, we have these 2 sites or 6 transects where Starry stonewort really took over, but we're not able to say what happened because we weren't able to resample them. So from conclusions of this work, we can say that Starry stonewort has a high capacity to impact native plants and alter the overall community. It's capable of affecting the number of native species present, the amount of native species present, and also has impacts to individual species. It's also altering the overall community. So when Starry stonewort comes in, it can be a very different native aquatic plant community than before. What you can expect when Starry stonewort kind of establishes and invades is that there will likely be significant declines in native cover and losses of a few species locally. So having a big change on the native community. And what we saw from this resampled transect data is that Starry stonewort readily displaces native species and can grow and expand rapidly, but lakes are different. We saw somewhat of a different response in moose that potentially is a higher diversity lake, is more resilient, but still in Moose Lake, Starry stonewort's doing very well in some places because we weren't able to locate those couple transects. So these conclusions really augment the body of knowledge we have on Starry stonewort. There was one published paper on the impacts of Starry stonewort that looked at total richness and total abundance effects, but now we have some more capacity to say how Starry stonewort affects species and groups of species. Takeaways from this work is that early detection of new populations are really important because once Starry stonewort establishes in a lake, it can grow quite rapidly and expand across the lake littoral area. And in the lakes where starry stonewort is broadly established, like Coronis, Moose and Winnipegashish, we're seeing big changes in the native community. So if we're able to catch new infestations early, those have proven not as difficult to contain, and potentially impacts can be contained in those lakes as well. And I mentioned the goal of this research was to get a better sense on what is the threat of starry stonewort and how is it impacting our lakes. Based on these results, starry stonewort is likely a high impact aggressive invader and warrants the prioritization that management agencies, MACERC, and the public are giving it. So hopefully we can keep our eye on starry stonewort to prevent it from becoming a widespread problem in Minnesota, since it's still very early on in its invasion. And this work definitely informs the threat of this species in lakes. I'd like to say thanks to my funding through the Minnesota Department of Natural Resources and also the Larkin Lab here at the University of Minnesota. Dan has been my advisor, and I really appreciate his guidance on this project and all the field support I received to get this work done. Also from AIS detectors who helped out with some of this work. That was a fabulous time. So I'd like to thank everyone for listening as well. I hope you learned something new about starry stonewort, and I'm looking forward to answering your questions.
All right, thank you, Carly. So we'll get the video started up here soon, and we're going to start working our way through the questions. We have had quite a few come in during the talk, but as you have them, as a reminder, just go ahead and continue to type those into the chat panel, and we'll work our way through the ones that we can. Carly has also kindly offered to respond to questions that we aren't able to get to during the webinar via a document. So we'll type those up and send it to her, and she'll type up some answers for us. And we'll send that out along with the link to the recording when that's available. So again, chat feature is there if you have any questions. And we have just about 30 minutes to start working our way through the Q&A. So I'm going to go ahead and get that started now. The first couple of questions that I have were just relating to the study design. We have one person wondering if the regression data that you presented was also over a 3-year period, or was that a single season?
Yeah, thank you for your question. So the data I presented in the first part of the talk was space for time data. So it's just transects that were set up one time, sampled, and then that's it. So the graphs that I showed you with the 3 lake lines, those are a single snapshot in time of those transects, looking at all the data that I collected in those lakes at the single point in time.
Great, thanks. And one other question along there was wondering if the research had any controls or accounting for water depth, clarity, nutrient, or other variables like that.
So another part of the sampling that we did that I didn't mention because I just wanted to focus on vegetation is looking at environmental conditions on these transects. So we sampled depth, we looked at sediment conditions, including texture, kind of nutrients that are in those transects, and we looked at that and it didn't have any effect on starry stonewort abundance. And when we set up the transects, we tried to control for environmental conditions as best as possible.
Great. All right, so we have a couple questions about control as well. So one of the questions was if there are any treatments that were being conducted on your study lakes during the time that you were studying them.
So Coronis was the only managed study lake that I had, but since starry stonewort is so broadly established in that lake, there's ample habitat that's not treated, and we work with the Lake Association to make sure all of our sampling was well away from any managed area. So we do not have any confounding variables of management in that data set.
All right, so is there anything that can help impede the growth of starry stonewort or effective control tools?
As far as control for management, it seems like there's good practices for controlling the biomass of starry stonewort using various copper-based algicides. There's been a lot of success in reducing the nuisance biomass, but carryover control, that's more uncertain, and that's likely to do with the bulbils of starry stonewort. So that's kind of one area of research on starry stonewort management, is how can we get good long-term control.
And hi everyone, this is Dan Larkin, Carly's advisor and a colleague of Megan's in AIS Extension. And one thing I wanted to add to that is that there have been some cases in Minnesota where early infestations have been found early on when they're still quite small, and there have been some promising results of hand removal of these very small infestations, which are quite different than the really the lakes Carly was working in, where there's really extensive populations spread over larger areas of the lake.
Great. Thanks, Dan. We had a question about controlling small populations, so very timely. Okay, so there's a couple of questions as well with other observations that you might have had during the study. And so some of the questions involved if there area, or why you might think that some areas at Moose Lake might have been more resistant or had lower populations of starry stonewort.
So for Moose Lake, it's definitely a higher diversity lake. I think in that lake alone, we found 20 unique individual native species, compared to Coronis. I think we found 13 native species. So in that lake, you're just having a higher average diversity and abundance of native plants. There's been some work to show that potentially a good, healthy, diverse native community can make it more resilient to invasion. So we didn't have any explicit tests looking at how diversity would affect the invasion. It's just kind of an interesting point that we saw as we were sampling these transects, just monitoring them over time. But Moose is definitely a higher diversity lake. It also has better water quality, so potentially the native species are more competitive in that scenario, or as competitive as starry stonewort.
All right, and then so some things that might have been outside of the study, but maybe some anecdotal evidence you might have. There's a couple of people wondering about impacts to fish. If you know anything, if there can be impacts to fish because of starry stonewort invasions, or if you noticed anything during your studies along those lines.
So I didn't look at impacts to any other trophic levels. Those type of studies are a lot more difficult to make clear conclusions on the impacts of an invader, but I wanted to get a really good sense of how starry stonewort was affecting native plant communities, because those are kind of the foundation of lake ecosystems, and you could potentially make some hypotheses on how changes to that community could affect others, but there's no evidence, or no one's looked at impacts to fish. But I did see plenty swimming around me when I was scuba diving. So.
And do you know if starry stonewort can have any impacts on other aquatic invasive plants, like, for example, populations of Eurasian water milfoil?
So all of my transects did not have any other invasive aquatic plant because I didn't want to have to deal with that type of confounding variable, having another invasive species. So I didn't look at invader-invader interactions, but certainly starry stonewort is in lakes that also has other invasive aquatic plants, and it would be interesting to see how they interact as a community.
Great. And let's see, did you notice if there are any significant changes in things like water quality within your study lakes during that study period, or water clarity?
So we only sampled water quality or water parameters at the single point in time when I did that broad sampling effort across 3 lakes. But we did see that there was kind of minimal variation between water clarity, at least within lakes. And we did some looking at if that affects starry stonewort abundance at all. And we weren't able to say anything about how kind of fine scale variation in water quality parameters affected starry stonewort abundance. So it was pretty consistent water quality data.
And then did you notice if there were any transects during your study that started at 100% native plant cover, and then by the end of the transect had transitioned to 100% Starry over the course of your study?
So that was the ideal goal of the transects, and we tried to set them up as best as possible to be completely uninvaded and invaded. There's definitely some transects that had that clear transition, but we were also trying to control for other environmental factors like depth. So we did as best as we could setting up transects that had really good native coverage and Starry stonewort coverage.
All right. And do you think that the longevity of the invasion each lake, so for example, Coronis having been one of the lakes that's had Starry stonewort longer than some of the others, might have some impact on the results that you're seeing or the differences between the lakes?
So we can't say for sure when each lake was identified. When Coronis was identified, it was already across, dozens.
200 acres.
Yeah, 200 acres. So could have been there for a while. And then for Moose Lake, it's also across a good portion of the lake as well. So we can't really say for sure if Starry stonewort invasion is progressing faster in Coronis because it's been there longer. But certainly coverage area suggests that it's had a good period of time to grow in that lake.
And I think there's also, in addition to time since invasion, as Carly alluded to before, there are probably some differences among the lakes in terms of environmental conditions that affect how well Starry stonewort does relative to native species. And so for whatever reason, Coronis is an example of a lake where it really seems to thrive and grow really abundantly.
And the good thing about these permanent transects that are set up, when there's resources available in a year or a few years, those transects can be revisited to see what they look like after a couple years. It'd be interesting to see if Starry stonewort maintains its dominance or if it was kind of a boom phase where it's really taking hold in a lake and then it kind of tapers off, because we sometimes see that with invasive species. So these long-term transects are still in the lakes, and hopefully they might get resampled in the future to see how the invasion plays out even on a longer time scale, more than 3 years.
Great. So I have a few questions for you now, more about Starry stonewort biology or ecology. So one of the questions was wondering if Starry stonewort persists through the end of the season, or does it have a dieback during the fall and winter?
So for phenology, we've looked at the phenology of Starry stonewort and also Coronis and Moose. These are kind of common study lakes for us. And we've seen that Starry stonewort kicks off its growing season a little bit later, not till early July we've seen in lakes. And this is consistent between Coronis and Moose, even though Coronis is very central and Moose is higher north. And then what we've seen is it really takes off in July and it continues growing and putting on biomass through October even. We've seen peak biomass occur late in the season, like October. And we have sampled Starry stonewort under the ice a couple times, and we see that it maintains some level of biomass throughout the year. It doesn't look great under the ice, but sometimes there'll be some green patches, and for sure just kind of laying along the lake bottom. So it persists somewhat throughout the year.
And the baubles, in addition to being a means by which Starry stonewort can spread to new areas, are also basically structures for hibernation during the winter. So the Starry stonewort will produce more baubles after it hits that peak biomass. And then those baubles will overwinter in the sediment. And then, as Carly was saying, when things warm up in the spring, you'll have a flush back. So it's a way. The baubles are a means of spread to new areas, but also of, sort of cycling within an area seasonally.
So would you say then that there's not a period of the year where there's no risk for transmission of Starry stonewort? There's viable pieces year round that could lead to spread?
We've seen baubles present throughout the year. I'm not sure how viable the material is right after overwintering, at least the vegetation, but for baubles, we've consistently found them throughout the year. And from when we've done some lab trials with baubles, we've seen about 70% or greater sprouting. So they're quite viable structures.
There is some difference seasonally in terms of spread risk from boater movement. So, once it's really grown up through the summer and hit that peak biomass and is closer to the water surface, that's when you're more likely to have material get stuck on props, get chopped up and potentially spread within that lake or taken to a new lake.
Great. Sorry. So does Starry stonewort fix nitrogen? And if so, could it be something that leads to an invasion meltdown, making conditions more suitable for other invaders like Eurasian water milfoil or clearleaf pondweed?
Sure. It is not a nitrogen fixer, but charophytes, Starry stonewort, and others can definitely influence nutrient cycling. And so it's possible that there could be some effects on nutrient dynamics from Starry stonewort. If you have this production of a lot of biomass and then there's sort of dieback and senescence, nutrients can be released, but there's not nitrogen fixation specifically. So it would be sort of similar to what you might see with any aquatic plant that produces a lot of biomass and then dies back.
Great. So are there also conditions, for example, nutrient richness that can make lakes more suitable for Starry stonewort?
Mm-hmm. Some of the lake level suitability modeling that has been done shows Starry stonewort tends to be present in lakes that have elevated conductivity and pH. So that's kind of the lake level studies associated with the presence of Starry stonewort. Beyond that, if you have any.
Yeah, no, Carly is exactly right. We've done work looking at sort of habitat suitability at a lake level, what lakes are most likely to get it, and those are the conditions. It's that sort of more conductive, harder water lakes seem at greater risk for Starry stonewort invasion. But one of the things we were really interested in, and that, as Carly mentioned before, she looked at, was getting a better handle on within lake invasion dynamics, and is that responding to environmental conditions and nutrient availability? And we, as Carly said, we didn't really see any clear patterns there, but that doesn't necessarily mean there aren't patterns to be found. It was one season of sampling looking at certain sediment parameters, and it may be that we just sort of missed some of the key factors that might be there.
Great. And then I have one more question about biology or ecology, for now at least, unless more start coming in. So this person had looked at sampling on Lake Winnie over the summer and saw that Starry stonewort seemed to be replacing Chara in some of the more shallow water, but seemed to also be colonizing areas that hadn't had plants in the past. So the question was, is there potentially an ecological benefit to Starry stonewort in stabilizing substrate or any other ecosystem service?
Yeah, I'd certainly say with any invasive or non-native species, it's not always bad and not all bad. So, I mean, depending if there's a lot of bare habitat and someone values having some vegetation there to stabilize the sediment, say Starry stonewort is pretty hardy. What we found is that it tends to grow more in mucky and nutrient-rich sediment. So I don't know how readily it would colonize kind of poor or coarse habitat. But certainly across a lot of lakes, we're seeing Starry stonewort easily replace Charas as the dominant species. I think as of this year, in Coronis, Starry stonewort has now displaced Charas as the dominant species, and that's at a lake-wide level. So it certainly is replacing out that Chara. And as I mentioned before, we talk about Chara as kind of just a lump group, but it is multiple species, and there's some rare species that we found in Minnesota lakes, so having impacts to that group is a big deal. It's a negative, it's unfortunate.
Speaking of Winnipegaushish, do you want to comment on what you saw of the Starry in Winnipegaushish compared to the other lakes?
Mm-hmm. Yeah, so in Winnipegaushish, I mentioned it was a flow through for the Mississippi. It's basically widening of the river channel. It's very sandy, not very mucky or nutrient rich, and across all the sampling, Starry stonewort was pretty low abundance there, especially in the shallower areas, which are likely exposed to more wind and wave action. So Starry stonewort doesn't really have any true roots. It has some clear filamentous rhizoids that offer some loose connection to sediment, but it's not going to be taking hold very well. So in the shallower areas of Winnibigoshish, it definitely was not growing very abundant. I wouldn't even call it a nuisance there. But I've also talked to some people that work on the lake there, and in the deeper areas, starry stonewort, I guess, grows a little bit more dense, which makes sense because they're more sheltered, not as exposed to the wave action. So certainly, starry stonewort is behaving differently in lakes, even just within Minnesota and across its entire range in the Great Lakes region through the Northeast. I bet it looks different. So it's always interesting to hear accounts of how starry stonewort is behaving in different lakes.
So a follow-on question to the charophytes was someone who noticed that starry stonewort is typically mixed in with the native chara in lakes where it's found, and they're wondering if it was difficult for you to differentiate the two as you're going through your transects.
Mm-hmm. Yep, yeah, certainly we see a lot of mixing of starry stonewort with charas as well. We don't know if that's kind of an intermediate stage or if the chara and starry stonewort are able to persist over long periods of time. But actually, when you're underwater, it's quite easy to distinguish the two because all the charas have calcification on them, so they look rough, they have texture, whereas the starry stonewort does not have the calcification. It's very smooth. So we feel confident that our cover estimates of starry stonewort and chara are good even when they're mixed together.
Great. And then I have a couple of people who are wondering if you could talk a little bit more about how starry stonewort is spread.
Mm-hmm. So starry stonewort can grow from an individual fragment. So the body of starry stonewort is kind of a stem and then whorled branchlets and a stem. And between each branchlet is just one cell. So if it gets broke, the whole cell all the fluid comes out, which is an identifying characteristic of Nitellas. So to grow via, just a fragment, it has to have an intact piece of a cell. It has to be like one entire branchlet, and then it can potentially regrow. It doesn't fragment itself like milfoil does, where it just kind of sheds off a stem and that can grow. It has to be an intact piece. And then certainly the bulbils are definitely contributing to the population growth of starry stonewort and potentially the spread.
And one interesting thing about it is this is a species where males and females are separate individuals. And to this point, only males have been found in North America. And so what that tells us is there's not sexual reproduction here, at least that we know of. And therefore, all the spread is by these fragments that Carly was talking about and bulbils. And we also see a lot of association with accesses and kind of other features of boater movement. And so it's likely that the majority of the spread is being driven by boater movement and people picking up fragments and bulbils in various ways.
Great. So did you notice if there are any depths at which starry stonewort was less successful or did not persist, or areas where there was less light penetration? Any limits in that area?
Mm-hmm. So we saw in Coronis, at least, we saw pretty abundant growth. Our transects, I will say the broad transects, the deepest depth that they went was 12 feet. That's just looking broadly across all lakes. And starry stonewort has been known to grow deeper than that, so we didn't sample in very deep areas, but certainly in shallow areas, if there's not a lot of wave action, you can have starry stonewort growing up, filling the water column. We've seen mats that are easily 3 to 5 feet deep, so it can grow quite tall, even though it's kind of a small individual. It just grows up together in mats. But I will say, the most abundant we've seen it is anywhere from 2 to 9 feet. So it seems to be very tolerant and able to grow successfully in a broad range of conditions.
Great. So along those lines, do you know of any indications about what climate extremes, for example, the extreme colds we've had this last year and potentially predicted for this year, might have on starry stonewort's ability to thrive in Minnesota?
So you've done some climate forecasting, correct?
Yeah, we've done a little bit of work trying to model the effects of climate on starry stonewort. I think it's likely that year-to-year differences are affecting growth of starry stonewort. So, you know, kind of accumulation of heat or growing degree days, you know, in spring and into summer probably affect year-to-year variability in abundance. But I think it's unlikely that those types of extreme winters or anything would make it so that Minnesota is not a hospitable environment for starry stonewort. For one, we see it in places like Red Lake in northern Minnesota that have, you know, are in a pretty extreme climate. And then also, it's really different in aquatic systems than it is in terrestrial systems because, you know, once you have that ice cover, the temperature that stuff under the water is exposed to is really buffered. And so you get down to 4 degrees Celsius and it's not going to get any colder. And so whereas invasive plants on land could potentially be more vulnerable to those types of extreme winters, invasive plants in the water have that sheltering effect of the buffering of the water and the ice cover.
Great. I think I have time for one more question. So did you, during your study, do any water chemistry analysis or track any of the water chemistry parameters alongside it?
Yep, so we have a water chemistry probe that we bring out in the field, and with that we look at pH, conductivity, dissolved oxygen, and temperature. So those were the 4 main water quality parameters that we looked at. And in terms of lakes, water quality or chemistry parameters are pretty consistent across the lake. The differences are when we look at different lakes, for example, Moose and Coronis, those definitely have very different water chemistries. But the modeling that I did to see if starry stonewort abundance was affected by any of those water chemistry parameters was kind of null. We weren't able to say anything about how it was associated with starry stonewort's abundance.
One factor, just kind of getting into the weeds on that a little bit, that could complicate things is that, as Carly described, she focused on measuring abundance using cover, but starry stonewort shows a lot of kind of plasticity in its growth form. And so it may be that future research looking at biomass as a response rather than cover could perhaps be more sensitive to these environmental influences, because you can have places like Winnipegoshish where maybe there are areas that have a really nice carpet of starry stonewort, and so you get those high cover values, but have less total biomass and less height of starry stonewort. So biomass perhaps could be a way to pick up more of those relationships to nutrient availability, sediment conditions, etc.
Yeah, so for the plant-to-plant interactions, cover was definitely the right way to go because we were able to sample broadly, and that has been shown to be correlated. The cover of native plants is equitable to if you were sampling the biomass, but in terms of trying to identify and tease out what's associated with these really nuisance growth levels of starry stonewort, potentially that could be looking at biomass as a better response. And
then there are also other elements that one could look at that we didn't have in this research, like calcium, magnesium, or other plant nutrients.
Great. Thanks so much, Carly, for taking the time to share your information on the webinar. For those of you still on, thank you for joining all of us. A reminder that we did record this and we'll have it available on our YouTube channel. You can see the link there to go ahead and subscribe. We'll also send out a message via Eventbrite to all the people who registered with a link when that's available. I know we weren't able to make it to quite all of the questions, so Carly will work on getting those, and we'll have those available once we post the link to the webinar recording. You can see on the screen contact information for Carly if you have any questions that you'd like to direct towards her. If you have questions about our program, other webinars, or work like that, our program email is there, and we'd be happy to respond to you there. So thanks again for joining, and we hope you found today very informative.
Thanks for watching.